So indeed, I'm going to talk about a computational framework for hypothesis testing in bioreactor biomechanics. And I'm speaking on behalf of the Cultivated Meat Modeling Consortium. I'm Jaro Camphuijsen, and I work at the Netherlands eScience Center, an expertise center for scientific research software. So here we see a picture of the CMMC, the Cultivated Meat Modeling Consortium, in 2019 at its inaugural meeting. There was a bunch of happy people there. And the consortium was supported by 2 companies. And in the meantime, we grew to 82 members. We don't have a group picture now because of the current situation, but we would love to have one at some point. And we now have lots of organizations that support us, either in kind or in cash, and we want to grow even more. So we have a couple of projects going on, and I want to talk about a couple of them. But first, let's talk a bit about computational modeling and how we see computational modeling and its value to cultivated meat. So here you see an abstract visualization of the engineering and science field, and we think that computational models are really in the middle. They're like an encapsulation of knowledge, and not only an encapsulation, but actually an actionable encapsulation, where you can take the knowledge that is maybe from literature or from experiments and put it in an actionable model with which you can do virtual experiments. So on the science side, we have this iterative loop where we build the model. We take knowledge rules from literature or experiments, implement them in our computational model in the form of mathematical functions or so-called knowledge rules. And then running a simulation with this model, we can reveal certain implications. And these implications we can again compare with lab experiment results to validate that our model is actually true. And most often this is not the case. And then we go back to the knowledge rules, improve them with the new results from the experiments, improve our model and do a new simulation. And this iterative process goes on and on until you have a model that is predictive and validated. And with this predictive model, you can do then simulations in totally new parameter spaces and geometries, for example, in the new bioreactor design that's not tested yet. And you can do predictions about these new setups. So these new simulations you can compare with, for example, desired product qualities. So here the product can be a new bioprocess or a new bioreactor design, or even just the actual meat. So we optimize for a certain parameter, be it maybe an optimal amount of cells, maximum amount of proliferation. And in this way, we can optimize our process. Often we find that if we actually implement the findings from these engineering models, we need to go back to the science side to improve because we get different results again. So this is this double loop that we go through in building models. So the first project we started with the consortium was the proof of concept model, which was funded by Merck. And we started with these knowledge rules. And for this particular project, we started with this really simple bioreactor geometry, the spinner flask, which is widely used, and some simple biomechanics. So this was really a proof of concept. So we really took, the basic knowledge rules that was needed. So we fed the bioreactor geometry to COMSOL, which is a fluid dynamics, computational fluid dynamics framework, which outputs a fluid velocity field, so a three-dimensional fluid field in time. You see a slide here. And together with this cell biomechanics rules that we took from literature, we fed that to BioCellian, which is a agent-based model, agent-based modeling framework, which models each individual cell, in this case on microcarriers in the bioreactor. And from these knowledge rules, we got certain behavior of the cells. So for example, if they experience stress, in this case, they turn red, but they also stop proliferating in a certain way. And you can, for example, also add behavior for cell attachment and all these kinds of things. So biocellian outputs then cell count, number of cells, but also their positions, the positions of the microcarriers, and the stresses that all these cells experience. And then this data is fed to a Unity 3D application, which makes this awesome interactive visualization. For that, you'll have to wait a second. It's terrible suspense because I also want to say that all this software is available on GitHub to look at and to contribute to if you want to. Now to the after the terrible suspense, let's look at this interactive visualization. We have a short video here, but you can actually access the visualization on our website, cmmc.org/stirtankdemo. So here you see the bioreactor and the spinning rotor, which is simplified but effective. And when we zoom in, we can see the actual microcarriers with the cells on there, which float around in the fluid, the medium. And you can zoom in on one of these microcarriers and see how these cells proliferate on the microcarriers and how they experience the stress through their contact with the microcarrier and the other cells, but also through the fluid velocity field and the shear stress. And on the right, we see a graph of the results of this simulation for two different stir speeds. So we see that for a very high stir speed of 240 RPM, we have significantly decreased cell growth. The full technical report can be read on Zenodo, and I'll link it also in the Slack. So now we go on with the next project, which is funded by the Good Food Institute. While the proof of concept project still goes on, we keep improving the model, but we also want to model all different kinds of bioreactors in this new project funded by the Good Food Institute. So we start, for example, with a rocking bed bioreactor that is used by Celtainer. We work with them to model this. Also the hollow fiber bioreactor and the continuous bioreactor by Cellular Revolution. And for all of these, we want to model the bioreactor with the cells in there and look at the necessary biological processes that these cells experience and at the geometry of the bioreactor and see if we can optimize for optimal cell growth. So this proof of concept was really nice and illustrative, but it's not yet very useful because we want to have more information about what these cells experience and about their behavior, why they behave like they do. And this is not yet encapsulated in the model. So we want more information, more behavior of these cells, more biomechanics and biological behavior. And then you can think of all kinds of things. I was very interested by the talk by Ben. I think we should put it all in the model. But then if you do that, then it takes like the age of the universe to run your model because of the computational load of putting all this biology in. So we have to make choices. And we decided to, for now, in this project, go to a simpler system, so not a full bioreactor, but zoom in on just a plate with a couple of cells and see what the effect is of shear force on the cells, and see how they behave if we increase the flow speed, for example. So how many cells will die, how many cells will stop proliferating, and there's a whole wish list of stuff that we want to know about these cells and that we want to experiment with. But again, we need to focus on what's important here. So we are also interested in your opinion there. Then again, we want to apply this information. So we make a model for this cell, and we want to apply this in the bioreactor environment for these different bioreactors. so we also need to know how, for example, the turbulent flow, which is present in the bioreactor, influences these cells. And because we're a modeling group, we're software developers, we don't know what's possible in the lab. So we started having these experimental design meetings with lots of labs from academia and also commercial labs, which was great fun, but also very informative. And we had a lot of very nice outcomes there. So we're still keeping up with this work, and we're designing new experiments, and also together apply for new funding to do these experiments in combination with computational modeling. And now this is my last slide to say, what can you do? Well, normally we say, go to the website, try out the demo, read a white paper, blah, 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 blah. But because this is such a really nice scientific audience with lots of expertise that is really interesting for informing our models, I would say just email me because we're very interested in working with you. And I have this very ugly arrow here to make this extra clear. We would love to work together. Thank you. This was it. I would welcome any questions that you have.
Thank you, Jaro. There are no questions on the Q&A. I don't know whether there are any in the Slack. They may come a little bit later. But I guess the obvious question is, this is extremely complex, this modeling work, and you already mentioned if you want to kind of approach the biology, you need computational power that far exceeds what we currently have available. So, for instance, the rather simplified kind of microcarrier system, when do you expect that this modeling will truly kind of reduce the amount of experimental work that we need to do to optimize these systems?
So, besides really predicting actual lab results, what we noticed is that it also really helps in actually formulating research questions. And I think that might be even a more important result than the actual predictions, because it helps also experimentalists to think about what's important, what they want to look at. So that's already helping in advancing the experimental work, I think. And then from a really practical point of view, I think in a half year, we can start doing actual experiments that start to matter, that can replace experimental work.
Right. You actually had that in the title, so maybe it was an obvious question. Yeah.
Right.
In the meantime, there's one question from Maria Olenik, and she comments on the spherical nature of the cells on the microcarriers. Are they actually modeled to be spherical, or are they single points?
So yeah, that depends on how you look at it. They are modeled as a center with a radius, so they feel each other's presence with a radius. So it's actually a sphere. This is of course very limited, and we're also looking at different cell shapes because probably deformation is also very, very important for how these cells behave.
Yeah, and when they adhere to a microsphere, they are flattened, right?
Exactly. Yeah.
There's another question from Varsha Rao, and this is an obvious one, I guess, is from the work on the 3 different bioreactors, or the 4 different bioreactors, and from the very preliminary data. Is there already one that stands out?
No. We haven't applied the whole model on these 3 other bioreactors yet.
Okay.
Also, I want to press that this is not like a race. We're not trying to pick out the best bioreactor. We're trying to optimize all of them.
Right. But obviously these questions will come up very quickly because everybody is dying to know these things. But Varsha, you have to wait for a little while. All right. Well, thanks a lot, Jaro. And again, great work. And please, everybody who wants to know more about this, contact either Jaro or look at the website of CMMC.